All right, guys, now that we've talked a little bit about the history of microbiology, let's kind of get into the nitty-gritty and look at the cell structure and the morphology of some of these prokaryotic cells, these bacteria that we've been talking about. And this will start to match up with some of the stuff we've been doing in the lab section of the course, okay? So let's start with scale, all right? So here in this this graph, you can see how it is that what we can see with a light microscope, and you can see the level that we're at with an animal or plant cell. Okay, we're talking on the order of 10 to 100 microns. All right, and I'm pretty sure many of you have seen red blood cells, which are some of the smallest mammalian cells, and they're down a little bit below 10 microns, all right? Obviously, we need a microscope to see these, and we need to go even into more resolution and more magnification to see some of the subcellular structures of an mammalian cell, like mitochondria. But this is the level, the same level that we're at when it comes to bacteria. Okay, so some of the subcellular structures of mammalian and plant cells are on the same scale as the bacteria. Now, we've talked about how it is that prokaryotes actually include bacteria and archaea. Okay, we talked a bit about the fact that archaea were some of the archaic forms of these unicellular organisms, and they set the stage for other prokaryotes, right? So archaea were some of the earliest prokaryotes, and then bacteria were some of the later prokaryotes. Now, if we go a little bit lower here, we have quite a range when it comes to viruses, okay? And we are going to talk about viruses. They are going to have a good section within the course, but a good part of what we're going to talk about here first will be about bacteria, about the prokaryotes, to some extent archaea, but mostly the bacteria that are part of that group, okay? All right, so let's kind of just get a synopsis of what microbes would include. Microbes would include the prokaryotes, both bacteria and the archaea, but it would also include eukaryotes. So some of these organisms will not get as much attention in this course. Fungus, fungi, including yeasts, will get some attention, but not as much. Protozoa and algae, these are all technically considered microbes, even though they're in the eukaryotic group, okay? We're going to start with prokaryotes and then go into more detail about what's making up prokaryotes. Now, we can talk about viruses, and we will in this course, as I mentioned, but viruses are technically not considered a living thing the way we define living. They're technically not a cell, though they do have genetic material within them and they do replicate. So it kind of stresses and kind of strains the definition of a living thing, but we'll be looking at viruses too in this section. All right. So first, let's take a look at the shapes of prokaryotes. In lab, we have seen that we can start differentiating between prokaryotes, between bacteria within that group, by looking at colony shape, and we've seen images of that in the lab section. We can look at color, the ruffled edges of colonies, whether those colonies are raised or not. These are all things in your lab section, okay? We also have seen that we could stain these cells. So now we're going from a macroscopic level of looking at colonies to the actual shape of these cells when they're stained and looked at in the microscope. So we have three basic shapes that I'd like you to know about. The first is a cocci shape, or coccus for singular organism with this shape, and these are more or less spherical, right? So spherical cells, we're going to say, are coccus or cocci for plural. Okay. We also have rod-shaped cells. Okay, so these particular cells are rod-shaped, right? And sometimes you'll see these cells are really long, and other times you'll see that they're really short rod-shaped cells, like you see down here below. Okay, so that not all cells are going to be exactly the same. We have cocci, spherical. We have bacilli, which are rod-shaped, and then we have spiral-shaped organisms as well. Now, this is not all that there are. There are just curved, kind of curve-shaped cells. There's a whole variety of different shapes of prokaryotes. There are even groups and species of bacteria that we would say are pleomorphic. They don't just show one shape, they show more than one shape. So if we say that an organism is cocci or a coccus type organism, we might say that it's monomorphic. It has one shape with an entire species. But then we'll see that some species are pleomorphic. They show a variety of different shapes. Some that are more looking like cocci, some that look bacilli, and this is just normal for some of these groups. Okay. Another thing that we could talk about when it comes to these prokaryotes is how they arrange themselves when they're growing. Okay. Sometimes we'll see that these cells actually arrange themselves always in groups of two. So we talk about diplo. We have this diplo prefix in front of the word, right? If diplo is the prefix and cocci is the suffix, then, well, these are spherical cells that are grouping in twos, right? Like you see here with this example, all right? We also see cells will cluster up, right? Like grapes almost. So here we see something where we have staphylo in the prefix, staphylo meaning that it's a grape-like cluster, and then cocci, of course, which is spherical shape. And here you see some examples of that. And in fact, some genus names of bacterial groups actually use this as their name, right? So Staphylococcus tells you a little bit about that entire group, that entire genus group of organisms, okay? Finally, we run into streptococci or streptobacilli, where these organisms will group up in chains, all right? So here, let's switch colors here for a second. So here we see a bacillus-shaped organism, and it's linked up to another of its kind, and another of its kind, and these are kind of grouping in chains. This would be a streptobacillus organism in this particular image. Okay, so these are all things that we would be able to see with a simple stain like we've seen in the lab section, and also some of the more sophisticated differential stains will also show us both shape and arrangement of the cells. Okay. So check out, this is section 3.3 in your text. We'll show you both some of these cell shapes and some of the arrangements we might expect, right? I would have you focus on those that we talked about in the last couple slides, right? There are lots of different kinds of things here on these graphs, but you definitely want to have the ones down that we talked about on the last couple of slides, all right? Okay, now if we look at individual cells and start to pick apart these cells, what I want to do is to start from the outside and burrow our way in, okay? So if we start from the outside of the cell and we're working our way into the cell, okay? So as we work our way into the cell, we're going to hit a barrier to the outside, and this particular barrier, change to red here, so would be considered the prokaryotic envelope. It's the envelope of this cell. It surrounds the cell. It's made of several different layers. In this case, we see that there's a possibility of a capsule on the very outside, a cell wall, and a plasma membrane. Okay, so we're going to talk about these one after the other. And what I'd like to do is to start with the very outside, which can exist as a capsule, but that's not the only type of extracellular matrix, they call it, that we could find. Okay, so we're going into this prokaryotic envelope from the outside in, and what we talk about is the glycocalyx. Okay, so a more general term for this outside section is a glycocalyx. Now this tells us something, this word glyco, and some of you might have run across in your chemistry assignment something to do with glyco or gluco that has to do with sugar. Okay, of course glucose is a sugar, right? So if you see glyco or gluco, we're talking sugar, and this is basically a sugar outside to cells that we often see. Okay, it's a sticky extracellular polysaccharide layer. All right, so this is part of your chemistry assignment. You had to look at different multiples of the monomers within a sugar molecule, right? So there's a monomer called a monosaccharide. Disaccharide would be two individual sugars bound together, and then polysaccharide is what you're running into here. Okay, so we have several monomers of a sugar that are making up this outside layer. Now sometimes cells will have what we call a capsule, okay, which is a more organized and firmly attached layer that's sticking on that cell wall. Okay, and here we have, actually, in this image here, we can see in white that there's this kind of diffuse kind of section that isn't taking on the red stain as well, and this would be the glycocalyx, and it may be that this is a capsule, or it may be a slime layer. Okay, so there's actually two different kinds of glycocalyces. A singular would be a glycocalyx, okay, that we might see on the outside of these prokaryotes. Now, what are the functions of some of these glycocalyces? One of the functions for a cell would be attachment. It allows a cell to attach to its substrate, so it can actually stick there, right? Now, so for some of these bacteria, that's the first step to infecting, right? So a cell can actually infect its host. A prokaryotic cell can infect its host unless it's able to attach to it. Okay, so to some extent, this is a first bad news for the host, right? Another thing that these outside glycocalyces that we see here in white around these cells will do is it will help the cell to prevent phagocytosis. So you may recall maybe in A&P or other bioclasses you've had that parts of the immune system, cells of the human immune system and a mammalian immune system would go around and gobble up these bacteria. Well, the capsule or the slime layer makes it harder for the immune system of an animal to deal with it. I have some other images here. So here we see Klebsiella pneumonia. Okay, now it kind of tells you one of the things that Klebsiella pneumonia causes, and that is pneumonia, though that's not the only thing that this organism could cause. But you can see here, this is actually a capsule stain, all right? And you can see kind of here also a kind of a diffuse outside covering to these cells, showing that there is a capsule associated with Klebsiella pneumonia. Down here in the bottom right, we see Bacillus anthracis. So you might have guessed that causes anthrax, okay? So here is the organism, okay? It doesn't look bacillus because I think it's just the slice that they've taken with this microscope slice with the actual sample, cut the rod kind of in that arrangement, right? It's not cutting it long ways, but it's cut it across. And you can see here that there would be actually a capsule associated with that Bacillus anthracis. Give you a couple other examples. Strep pneumoniae. Okay, so Streptococcus is the word in front of pneumonia for that particular species. So you might guess that, well, there's probably more than one organism that causes pneumonia, and that's why these organisms get that name, all right? So strep pneumonia actually makes capsules just like Klebsiella does. Another one, strep mutans, Streptococcus. Now that you were hearing these words, right, you're probably thinking, okay, strepto, chains, coccus, spherical cells, so these are in chains, okay, and mutans. Okay, so Streptococcus mutans is one organism that has a slime layer. It allows organisms to actually stick to our teeth, and it will cause dental caries. It actually causes cavities. All right. Again, kind of hitting home the idea that organisms that have capsules have a tendency to be more virulent, have a tendency to cause disease more so than members within its own species that don't have a capsule. Okay, so one bacterial species can have members that don't have a capsule and other members that do. Those that do a better job at infecting. All right. All right, so I think that's about it for now until we move onward and get to the cell wall. Okay, so right now let's call it quits, and we'll move more into that prokaryotic envelope and talk about the cell wall next. All right.